Risk factors of squamous cell carcinoma. Tobacco is a major risk factor. Tobacco is a source of intraoral carcinogen. All forms of tobacco consumption have been linked. It is either chewed or smoked. It is smoked in form of crude cigars or factory-made cigarettes. Tobacco contains nitrosamine, benzopyrene. All of these increase the risk of carcinogenesis and thus weaken the oral region immunity. IARC in 2007 stated that there is production of oxygen and nitrogen-free radicals. These radicals affect antioxidant defense mechanisms in the oral epithelium. Second major risk factor is alcohol. Excessive consumption of every type of alcohol raises the risk status of oral cancer. Also, it potentiates the effect of tobacco. It increases mucosal permeability, it causes irritation of the mucosa, and acts as solvent for carcinogens. The role of alcohol dehydrogenase is important because there is production of acetaldehyde, which is a cytotoxic molecule that fosters both the production of free radicals and hydroxylation of DNA base. The alcohol dehydrogenase type 3 genotype is associated with rapid oxidation of alcohol in acetaldehyde, which massively predisposes patients to oral cancer. Alcohol consumption is highly correlated with the development of oral cavity tumors and with the presence of various impurities like nitrosamine and mycotoxin. These are carcinogens. Alcohol abuse also significantly augments the effect of other carcinogens like tobacco. So there is synergistic effect. Cytochrome P450, a xenometabolizing enzyme, is involved in the activation of N-nitrosamines associated with tobacco. Betel nut chewing is an important risk factor. It causes some mucous fibrosis. Use of betel nut chewing that includes both tobacco and areca nut has been found to be 15 times more cancerous than chewing it alone. It releases reactive oxygen species that cause highly detrimental effect at the mucosa at oral cavity levels. These reactive oxygen species, ROS, takes place in alkaline conditions throughout autooxidation of the areca nut polyphenols in saliva. So there is increased genotoxicity and genetic mutation, and the salivary proteins are attacked. ROS causes structured leveled mutation of the oral mucosa. So there is penetration of other environmental toxic agents in the oral cavity. In North Africa and Middle East, a mixture of tobacco and lime water or oil is used. The presence of a correlation between viruses and oral cancer has been strongly demonstrated. Human papillomavirus, herpes group virus, adenoviruses, and hepatitis C virus are implicated in causation of squamous cell carcinoma. Human papillomaviruses are epitheliotropic DNA viruses, which induce hyperplastic, papillomatous, and verrucous squamous cells in stratified squamous epithelium of skin and mucous membrane. 16 and 18 human papillomaviruses are strongly associated with malignancy. These are often described as malignant oncogenic genotypes or high-risk genotypes. Oral squamous cell carcinoma has weak association with ill-fitting dentures, mucous fibrosis, poor hygiene, nutritional deficiencies, exposure to sunlight, and Palmer-Wilson syndrome. So to conclude, oral squamous cell carcinoma accounts for 90% of all oral cancers. Human papillomavirus and herpes virus are most studied and are considered those viruses which frequently cause human oral squamous cell carcinoma. Thank you.